sorry that Wally's not here. I'll probably take one. Thank you.
Well, I'll just put it down here next to you. So welcome everybody for the second part of our evening here. My name is Limor Joshua Tor. I'm a faculty member here at Cold Spring Harbor, and hope you enjoyed the play. I certainly have. First up, we have the playwright here, Keith Burgess, who's going to hopefully he is a professor at University of North Carolina, and hopefully he can tell us a little bit about his thought process and how he arrived at this play. And
Okay. Well, first of all, the play was intended for Jim's 90th birthday, which was in April, and we're 6 months late, but belated late. Happy birthday, Jim, and I'm sorry we didn't have it on time. So actually, let me start. I'd like to thank everyone for coming. I know some people have come a long way, and I really appreciate it. So thank you for coming. I'd like to thank Harold, the director, and Rachel, and Brad and Dominic, who I thought did a fantastic reading. Without a director and without the actors, there would have been no play, so I thank them. And particularly Mila. Where is Mila? Mila is there. So Mila, this was her brainchild. She approached me a year and a half ago and asked me whether I'd be interested in writing it, and she has shepherded it through the whole way, and so thank you, Mila. This is
the producer.
Yeah. I have one last thank you, which is to my wife, Pat Selling, who's sitting there in the audience, who read many, many versions, and thank you, Pat. When Mila asked me at the beginning, that was a challenge. It was exciting. I knew about the discovery. I knew from a little bit of the history, but I wasn't there. This is not a documentary, obviously. And so I initially did a lot of reading, and it's wonderful. There's some wonderful biographies. I first of all went to the 8th Day of Creation, which I hadn't looked at for 30 years, and I thought that was very good. But one of the wonderful things was I went to the 2014 mRNA videos, the meeting here on Messenger RNA, and the videos were wonderful. So many of you may have been here at the meeting and may have actually seen those videos, but for me that was great. And I actually lifted some things in the play, and some of you may recognize this from those videos. And I'm sorry that Wally Gilbert is not with us, because he certainly had some things which I sort of stole from. So for example, the Alfred Tissier story about him missing, not getting poly A to give polylysine, that was in his talk, and I didn't know about it before then, so that was really very informative. And there were other things too that I picked up from there. So in terms of the play, it was a challenge at the beginning, because I realized I couldn't do it as a documentary. I couldn't do it with somebody pretending to be Jim or pretending to be Matt or Wally Gilbert. That would never work. So obviously I had to invent a couple of characters, and that's what I did. And so in a way, I think the play, it uses the discovery of, which is a momentous discovery. It's such a huge, probably one of the 3 big discoveries in molecular biology of the 50s and the 60s. And I use that as a vehicle to think about, what motivates people. And obviously, curiosity is one of the things, but I talk about ambition and competition and jealousy, which is talked about in the play, and you can see that coming out between the 2 groups. And obviously that is something which I think any of us in science, we know that does motivate us and can drive us sometimes inappropriately. And so I think I was illustrating that. So at the beginning I had the play really with just the 2 characters, Alan and George. But then I realized it was really a little bit dull, just having the 2 old guys sort of reliving the past. And I think the introduction of having Anne as a character, I think maybe she's the most interesting character, but it also gave me a chance to talk about how difficult it is, obviously, for women still to this day in science. And certainly back then, I think it would have been extremely hard, and the choice between having a family and being pregnant and whether she could continue. And obviously, some of the rules have changed now, but there were certainly universities who would not hire. If the husband had a faculty position, the wife could not. And so I felt, that was a way to sort of, bring that into the play, which I think maybe it's something we may even discuss. I don't know. There were many things I left out. I wanted to have perhaps some of the things more with Sidney Brenner. Sidney, and many of you may know these stories, but when he was in Matt's lab and with Francois Jacob, and the experiments weren't working, and I think many of you know this, it was like one of the last days they go to the beach, and the experiments haven't worked, and then Sidney suddenly realizes it's magnesium, and he leaps up, it's the magnesium. The magnesium was too low. And when I was a graduate student, and so Sidney and Francis were the head of the floor that I was working on, and there was a coffee room. We'd often go into the coffee room, and Sidney would always hold court, but if somebody's experiments weren't working, he'd always say, "Oh, it's the magnesium." And I never understood what the reason for this was. It sounded like it was a good joke. Only when I was really writing the play that I got to realize, it was the magnesium for him. So that was one thing. But the other experiment that, or vignette that I loved in the story with Sidney and Francois, having had this insight that it was the magnesium, they rush back to Matt's lab to do one last experiment, and they're so nervous. And Francois is trying to inject the P32 into the tube, and he's shaking, and he misses the tube, and it goes into the water bath. And so then there's this screaming hot P32 labeled water bath, and Sidney takes the water bath and hides it behind the Coke machine. So I don't know whether you ever knew about that, Matt, because I think you did. But I wanted to put that in the play, but I couldn't quite see how to get that in. But there were so many other things. But anyway, so that's really how the play came about.
So one thing I forgot to mention is that you were an alum of Cold Spring Harbor as well.
Yes, I was a postdoc with Jim, and I thank Jim for giving me a fantastic opportunity. And some of the things that I said in the play, that you gave us great freedom, but I also felt some pressure. I sometimes worried that you were going to fire me. But you made my career, and I am very grateful, Jim, so thank you.
I never thought of firing you. But Lazaridis was another matter.
Well, there were a lot of characters there at that time. Yes, Lazaridis was a headache.
Yes.
And the parties?
Actually, the very first time I met Jim doesn't realize this, was at a party in the Golden Helix. I went to one party in the Golden Helix, and Francis, the reason Jim was there was that they were filming, so this was in 1973, I think, and they were filming, and it was for like the BBC and probably Nova, and so Jim had flown over, and Francis put up a notice very late on a Friday, party tonight, and only the graduate students really showed up. And we were excited because Jim was there, but the real reason we were excited was that the director of the movie, his girlfriend was Miss World. Miss World from, I can still remember, 1968. She was Eva Rubinstein. She had been Miss Austria, and so she was Miss World. So we were all keen because she was there. But Francis danced with her a lot. But the graduate students, we were not really considered. But that's when I first met Jim. I don't know if you remember going to
No, I don't.
Yes.
Parties and memories.
Yeah.
Cool.
No.
So amazingly, we have some of the original players in this story. And I can't believe that both of them are high school dropouts. I just learned this evening. And so we're going to start with Jim Watson, who is, of course, here at Cold Spring Harbor, and he'll tell us his take on the events. Jim.
Yeah. Francis generally gets the credit, at least in the literature, for saying DNA goes to RNA, it goes to protein, but I had put it down on paper in November of 52, that is before the DNA structure, we were close to it because of reading the literature at that time on, there was very little about DNA, and most of it was on RNA, and I would have to do research to find out what I was reading, but there were two principal characters, Jean Brochet in Brussels, and Casperson in Stockholm, and they were both made the observation that when in cells where there's active protein synthesis, RNA is very visible. So RNA comes up when you have protein synthesis and goes down. And the only way, and by then I thought, it's convincing DNA has to be the gene, and that RNA was, for some reason, an intermediate. Anyways, I wrote it down, DNA to RNA to protein, and then after we had the double helix, and I went to Caltech. That had been arranged before we had the DNA structure, and I was to be sort of a senior postdoc of Delbruck, and what I didn't know is Delbruck wanted me to run the phage activities while he moved on to his Phycomyces, I think. And, but I was a total failure then, because when I got to Caltech, I was only interested in how proteins were made, and I lost totally any interest in phage. And so the first thing before coming to Harvard, I realized I had to have an experimental lab, and then offered a job as a postdoc to Alfred Tessier, a Swiss biochemist who was in Cambridge studying oxidative phosphorylation in mitochondria. And he knew he was sort of going nowhere. And I said, you should come to Harvard and study ribosomes. Because the other thing about protein synthesis, wherever it was actively being made, there were lots of ribosomes. And I forget, but I'm sure there was evidence of radioactive amino acids being stuck to the ribosomes. And so Alfred said yes, and came after I'd been in Cambridge for about, at Harvard for 6 months. And then he began to purify the ribosomes, and then we made the observation of the 30S, the two subunits, the small and the large one, sedimented at 30 Swedbergs, the other sedimented at 50, and they came together in magnesium. So magnesium, in some sense, bound RNA together. Or we had, of course, didn't know reason to believe it was RNA as opposed to protein. But in any case, magnesium, only with magnesium did we have reproducible results. And that I had, my first graduate student at Harvard was a young Canadian, Bob Riceborough, who came from Cornell, and who at Cornell, his principal interest was ornithology. And he was making a big leap from being interested in birds to being interested in DNA. And again, I can't tell you how it started, but he essentially added P32 in short pulses to to cells, and observed that, and this was after phage infection. And after phage infection, RNA synthesis almost stopped, but there was a form of RNA made which was neither of the ribosomal RNAs, the 16S or the 23S, and had a sort of broad distribution. But there was a characteristic new form of RNA which carried out the phage-specific synthesis. And I forget who gave the name messenger RNA to it, but we did those experiments in a convincing form in January through March of 1961. And then it was in Cambridge, and for some reason I learned that Leo Szilard was in Memorial Hospital being tested as to whether he had cancer. The answer was no, but I remember seeing him, and he was the first person I told of the results that there was this special form of RNA made after phage infection. And I thought it was, very important. And Leo didn't agree and said it's probably virus specific and has nothing to do with the general way proteins are made, and so we should study it in uninfected cells. Now, I think possibly even beforehand I had written Francois Gros would he come to Harvard over the summer and study uninfected bacteria to see whether you could see the message there. And at that time I was often fed in the evening by Celia Gilbert, and Wally Gilbert's wife, and the daughter of the famed journalist Izzy Stone, I.F. Stone. And Celia, who sadly can't be here tonight, is a very remarkable woman in her own way, just as bright and clever as her father. And whereas Wally generally never said a word. And Celia carried forth, and I think actually Celia valued me being around because I talked. Wally thought, and I talked. And occasionally there would be Shelley Glashow or Sidney Coleman also for dinner. They were young theoretical physicists, all attracted by Julian Schwinger. And Wally had a position called assistant to Schwinger. And apparently in theoretical physics you moved up by being an assistant, so that someone could test their ideas against you. And but Wally, when he'd heard of the experiments we'd done of phage, and when Francois came, wanted to spend the summer doing experiments with Francois. The trouble is, it was a sort of total failure in that which afterwards we realized that there was messenger being made, but it was overwhelming in molar amounts by ribosomal RNA. And so ribosomal RNA was, we used to say, between 1% and 4% of the total RNA. And you never, so you could see sort of the outline of the 16 and 23, but there was mess on the site. It wasn't clean, and that was undoubtedly the E. coli message, but it was very hard to prove. And what I now forget, and the key fact is, did I ever tell Sidney Brenner of our results with the T4 RNA? And my guess is I didn't. A big mistake. But I think I didn't want to incite, get Sidney interested in messenger RNA. But during that time when Wally was doing the experiments, which didn't prove anything, Sidney had, and Wally could say, but had met Francois Jacob at the Cold Spring Harbor Symposium. And the experiment that they devised could be best tested in Matt's lab, and Matt can talk about that. And so Sidney went off to Caltech for roughly a month, and we didn't remain in contact at all. Francois Gros went back to Paris, and in Paris had an American MD from Harvard Medical School, Howard Hyatt, who tried to take Wally's place in the messenger RNA experiments. And they kept promising they'll, get real results the next month, but they didn't really. And it was, hearing that Sidney had a manuscript, which led us to realize we had to write a manuscript. And there were the Howard Hyatt and Francois, and there were 6 names from on our side. And Matt could say, but the experiment from Matt, it was just a nicer experiment and much more, easy, clean experiment. So he got the credit. But we deserve credit for finding phage RNA and the messenger RNA, and it was Matt and Sidney and Francois that got the other. Anyways, that's my side of the story, and my memory is really very bad now. That,
seems very good. It seems.
No, I'd like to know exactly what I was reading when I wrote DNA to RNA to protein on a piece of paper, and my guess is I was reading Brochet. For some reason, I didn't like Casperson, whereas, or there was just something about his papers which, he never acknowledged DNA. It was all RNA, and but Brochet was a jolly man and very nice man, and after Brochet died, Brussels. There was one very good person who continued in Brussels, but and Casperson, actually 20 years later did better work. But it was long afterwards, and so the answer is we discovered phage RNA, and Matt proved E. coli RNA, which we could have probably never got through the editor of Nature if we only had our experiment. But we should have published the phage just separate. But we didn't know how universal life was, or the mechanism.
Jim, you had some slides you wanted to show us?
Yes, it's you know. Yes, this was Harvard, which the biological labs, when it was dedicated, had these two giant rhinoceri, which are still there, bronze. And there's Alfred Tessiers in the back, Fred Goldberg, me, Wally, I forget her name. The one at the end, it was Pat Cowan. She had beautiful blue eyes. And I don't remember anything except her eyes. And for a while, she became Mark Potashny's girlfriend. And David Schlesinger is an early student, Gary Gus and Mario Kopecki, my secretary, John Richardson, Ed McConkey, and I don't recognize the woman, but we took those pictures every year, and the high point of it was climbing up and helping the girls climb up. Next one, these are the papers that came out of our work, and that's the Nature paper, and that's the Cold Spring Harbor Symposium. And then Wally, you know, always had a pipe in his mouth those days. And Tessiers was a great alpinist. He had climbed one of the really difficult peaks in the Himalayas. And yes. And I don't know where I met Francois Gros first, but conceivably at a meeting in Brussels. The next one. Yeah. That's when we got the Alaska Awards. And yes, need for a Kennedy victory. And I went bald really quite early.
Okay, yes.
Next.
Right. Okay, thank you, Jim.
Okay.
So Wally Gilbert, as you heard, was supposed to be here and is really sorry that he couldn't. But we have an excerpt from his talk from 4 years ago from the Messenger RNA meeting. So here's that. That was interesting.
Two experiments in 1960. The answer is very crudely, fantastically crudely. Jim and Francois and I were doing messenger RNA labeling experiments. Francois would have in his hand a 20-liter flask in which he would be shaking at room temperature a culture of bacteria growing in 2 liters. I would be standing there with 20 millicuries of P32. Jim would be standing there with a stopwatch. Jim would shout, "Go!" I would pour the P32 in. Francois would shake this gigantic flask, well, perfectly like that, and then we would pour it into a gigantic vat containing ice and azide, stop everything instantly, carry that vat into the cold room, spin down the bacteria, get several grams of bacteria, grind them up with alumina, make an extract, layer it, as Jim mentioned, on sucrose gradients. We actually, originally, we'd make the gradients in the warm because we didn't like to work in the cold room, so we'd start with a warm sucrose gradient. We'd each carry a gradient carefully over, put it in the centrifuge, spin the centrifuge, take out the gradients, each of us carrying a gradient over in a tube, putting it in the holder, puncturing the bottom, holding, sucking on the top, allowing drops to fall gently, collecting them in tubes, counting them, and counting in those days. Originally, we had only a Geiger counter. We had precipitated the radioactive material on little filters and little planchettes. We took them upstairs, and we had a Geiger counter surrounded by lead bricks. And you could open the lead brick, stick the planchette under it, close the lead brick, and sit there with a stopwatch and start the counter and the stopwatch and watch the dials go around, the electronics go around, stop at the end of a minute, write down the answer, go on to the next sample. You had to have a lot of radioactivity to actually get any results that way. And that was the original experiments that the three of us did in the sort of May, June period that year. Soon after that, we got an automatic counter that would actually count the planchettes. And as Sydney described, you would actually have a tape that would come out, and you could actually now count for 1,000 counts and get reasonable accuracy. And simultaneously, we learned in doing that keeping the bacterial extracts in the warm was not a terribly good idea because things tended to decay, and we began to work entirely in the cold room. The technology became gradually better and better in the course of the year. And in fact, the idea of labeling 2 liters of bacteria with P32 turned out to be a mistake because the bacteria had a phosphate buffer in the growth medium, and you could equally well label 50 milliliters of bacteria at a much higher specific activity and get exactly the same result. The result was dependent only upon the time of exposure and the amount of P32 that was there to begin with. So that by the time I'll show you any experiments, the technology had improved tremendously from the first ones. Our paper appeared in Nature, as Jim mentioned. Bob Riceborough did all of the T-phage, did the T-phage experiments. I did most of the gradients that were finally in that paper, or many of them. And the theme of that paper, as Jim mentioned, was first to show that the that the T-phage RNA behaved not like the ribosomal RNA, but appeared after a long period after any period of labeling, was associated with ribosomes, was separable from the ribosomal RNA, and that we could see in a pulse labeling in E. coli exactly the same type of RNA appearing. And so that's what's in this slide, in which you see the ribosomes, the RNA in here for a pulse label. Here are the ribosomes and the RNA from the T-phage. And we knew from the work of Alfred Tessiers that the active ribosomes synthesizing polypeptide did not come apart in low magnesium, but in fact ran ahead down here at 70S. We could do an experiment of the kind of actually isolating, purifying those ribosomes stuck together as 70S ribosomes in low magnesium. And this experiment simply shows that, one, you can purify, enrich by repeated centrifugation for this population of ribosomes, showing here a pulse-labeled message. And you could take this apart and discover that this this material writing the ribosome was that same pulse-labeled material we had seen earlier. It was a wonderfully exciting time. When you actually look back on the papers, they're a wonderful mixture of true statements and false conjectures. And it's the false conjectures that one generally manages to overcome over time. Thank you.
So we have a special appearance now. Sidney Brenner really wanted to participate, but as some of you know, he's quite ill. Nevertheless, he recorded a video for us just last week in Singapore, and so I invite you to watch the video. He has a bit of trouble breathing, so please excuse that, but Sidney Brenner.
So you know, there was a meeting in April 19 April the 30th, 1960, in my rooms in King's College, Cambridge, at which we gathered to discuss some questions about ostensibly expression of the conversion of DNA, of the DNA sequence into protein. Francis had been preoccupied with the ribosome for several years. And it was at that meeting, because I happened to realize and knew Elliot Volkin from Cold Spring Harbor much, much earlier on, but I realized at that meeting that the something called the Volkin RNA was effectively the copy of the gene in bacteriophage T4. Francois was present at that meeting in my rooms, and that evening, that afternoon, he and I got together and between us planned the experiment that was to take place much later. That experiment was to prove that the RNA synthesized from as a copy of bacteriophage T4 and had a different base composition from the RNA of bacteria. That was the messenger, although we didn't call it that was added to ribosomes. We then planned to do this experiment, having found out that Francois was going to Caltech to some time later, Francois was going to Caltech for a few weeks to give lectures. And the experiment consisted in showing the new RNA was added to the pre-existing ribosomes. And that was the essential idea of the experiment. We had decided we would do this at Caltech, where we could use Meselson's, who was then at Caltech, where we could use his isotopes, C13 and N15, to label the old ribosomes, and then show the new RNA was added to old ribosome. Very simple. We thought it would take a day to do. So eventually, later that year, we gathered at Caltech to do the experiment. But no matter how many times we did it, the ribosomes fell apart and we could not achieve it. Of course, you realize that in order to spin the ribosomes to sedimentation equilibrium, we had to spin them for a very long time. And the solution we used was 8 molar cesium chloride. Now, right towards the very end fact, we only both had that weekend and a few days grace after that. We went to the beach with Hildegard Francois and myself. And while we were at the beach, as you can all read in Francois' account of it, it suddenly dawned on me that the cesium chloride was a huge competitor for the 1,000 molar magnesium we put in. And so I suddenly realized we have to add more magnesium. And of course, I leapt up saying, it's the magnesium. And we immediately got into the car and went back and repeated the experiment. The experiment was then to do this with under 3 conditions of magnesium. It was 1 millimolar, 10 millimolar, 100 millimolar. And we had 3 tubes where we were spinning this, so we got them all into the machine and started to the experiment going. And after a few things such as the centrifuge broke down and the whole rotor was carried by me through the corridors, down 2 floors, so we could use Renata Dulbecker's centrifuge. We managed to complete the centrifuge. The original data, which I kept for many years, is in Cold Spring Harbor. You can see the tapes on which was printed out the radioactivity. And Francois and I sat there watching the nuclear Chicago printer slowly, and it went up. It was flat, and then suddenly began to rise. And of course, the ribosomes that's where the ribosomes were centered, although you could not see them. But we knew where they were because there were markers for this. At any rate, that, as far as we're concerned, proved the existence of this. I don't know who first called it messenger RNA. There must be, but it's never been found, a paper, a draft paper, was written by Francis on messenger RNA without any evidence as early as April 30th. I used to have a copy of it, but I'm afraid that it must have been lost. As a result of this, it then took us on return several more months for us to prove that new protein was also made on the old ribosomes. By this time, there was not any C13, but we found that N15 was enough to give us the displacement that we require. So a large number of experiments were done by me later that year. And then at the end of the year, 1964, Francois came to Cambridge for us to write the paper, which we did. That was the paper that was first submitted, and because Meselson was an author, Meselson got a copy of the original paper, which he showed to Jim. Jim got very excited and said that they had done the same thing and had come. And so I decided that it would be important to have both papers side by side. And Jim said, they've done all the work, they'll send me the paper. Of course, the paper took a long time to come. In the meantime, everything got very complicated because Jacques Monod and Francois were writing their papers, which also had, as I called it then, and still call it now, the retrodiction of something called Messenger RNA, as indeed. But the significant thing, and if you read our original paper, which appeared, I think, in July or June, when Jim finally got around to sending us what he did, you can see that ours is the absolute proof. And I think it was a very difficult experiment. And I don't think experiments like that have been done unto this day. There were other things, but as far as I can say, that is my contribution to the concept. So I believe I proved that it existed without any doubt at all. And if you want to take that as something, you can take it for what it is accounted for.
Thank you, Sydney. And of course, said Matt Meselson is here with us today.
Since Jim mentioned Leo Szilard, I can't resist telling you a story about Leo. I visited him several times while he was sick in bed with bladder cancer at Sloan Kettering. And at that time, Leo had a plan to bring better relations between the United States and the Soviet Union. He called it the Angels Plan. He realized that a direct meeting between Kennedy and Khrushchev would be a very high-level commitment to anything that was said or even understood to be said, and that would prevent exploration of new ideas. So he got the idea that some family member of Kennedy and some family member of Khrushchev, they could meet, they would be the angels. It would be Steve Smith, the husband of one of Kennedy's sisters, and it would be Alexei Adjubey, the husband of Khrushchev's daughter. So he wanted to propose this to Khrushchev. So he called Dobrynin, who was the permanent rep of the Soviet Union to the United Nations in New York, and said he needed to see Khrushchev. We knew that Khrushchev was coming to address the United Nations. That's the time that Khrushchev beat his shoe on the platform. And he was told Dobrynin he needed 2 hours with the General Secretary, and Dobrynin's, this of course is all from, I wasn't there, this is all from Szilard. Dobrynin said 2 hours is not possible, it is too long. So Szilard, typical of Szilard, said, okay, forget it. So then Khrushchev comes to town, and Szilard gets a telephone call in his hospital bed. The General Secretary will see you. Now, at that point, Leo didn't want to haggle how many minutes. If it was initiated by Khrushchev, Leo would go. So he went there, and I wasn't there, of course. This is, again, I must emphasize, all from Leo. But he brought Khrushchev a gift. And he, according to Leo, had this gift in each of both pockets of his jacket. And he gave the first package to Khrushchev and said, "Mr. General Secretary, this is a special kind of razor. I use it myself to shave, and I'm giving you one. It's very good. It's called a Schick injector razor. And it requires very special blades. And here I'm giving you a 6-month supply, and every 6 months I will send you a new supply on one condition." And Khrushchev, according to Leo, said, "What's that?" And Leo said, "No nuclear war." And now, according to Leo, Khrushchev says, and according to Leo, these are both little fire plugs of men, puts his arms around Leo and says, "Don't worry, Leo, when nuclear war starts, I stop shaving." So it has nothing to do with tonight, but it's a story that should not be forgotten. Just as this play talks about things that should not be forgotten, I thought it was a very humane and a really very sweet play. And I think that the playwright is trying to do something that we scientists have failed to do, which is this. There are many interviews with individual scientists about what they did or what they witnessed, but almost never are they brought together so that one could say to the other, "It wasn't quite like that. My memory is different." I once proposed to the NSF that they pay for a few meetings where people could get together who were together at the time of something interesting happening, and then they could correct or amplify on what. No, that's not the way things are done. Okay, well, then you have the historians of science. Maybe they would do it. They would go to each scientist back and forth. That's not as good, but maybe most historians of science don't study living science. They wait till the scientists die. That means they don't have to understand much about the science either. So I'm very disappointed in the historians of science. At Harvard, I don't think they ever came over to talk to you, Jim. We have a history of science department. Never have any of them come over to talk to me. They sit there and they study how the social ambiance may have affected Pasteur. Okay, so I think that the playwright and the actors are doing something that we should have done and still don't do. Now, more to the subject. So, the history of the unstable messenger has never been correctly written. So far as I have read, it begins a full 8 years before the messenger experiments, with Pappenheimer, Monod, and Cohn-Basier, who was the wife of Stanier. This was an experiment in which they simply dump in a lot of inducer to a growing culture of E. coli. And they measure the production of activity, beta-galactosidase activity, at various times. And the production is an absolutely straight line that extrapolates to the very moment that the inducer is added. Many times this experiment is repeated and repeated and repeated at the Pasteur. Nobody tries to interpret or even guess to what it means. The explanation which Monod had was a word called the organisateur, which is an excuse for saying we don't know what the hell this means. It wasn't until Pardee, in his first year, 1957, at the Pasteur, a young American from Berkeley at that time, did an experiment which is essentially the so-called pajama experiment, but not in order to find anything out about the genetics. What Pardee was interested in, and he's the sole author of this paper, and the title of the paper is The Transfer of Information from DNA to Protein. And what he did was the same experiment that had been done again and again and again before that at the Pasteur, with no interpretation, just a statement repeated many times in papers by Monod, that synthesis begins immediately at full speed. So Pardee, instead of plotting the amount of enzyme made in the experiment he did, which was not to add inducer, but rather to add male bacteria who carry the gene for beta-galactosidase into the female bacteria who don't have the active gene, and now to measure the production once the gene has entered. But now the males don't find the females instantaneously. They're wandering around looking for females. Art had the genius to plot time versus the square root of the amount of enzyme, on the idea that the simplest approximation to the time course of males finding females would be the first power of time. He got an absolutely beautiful straight line with loads of points on it, concluded, as all these other experiments, which had used not males and females, but just dumping in inducer, but the same conclusion, that synthesis starts immediately at full blast. Art gave it an interpretation, which was the obvious interpretation, which is if the enzyme's not made right on the DNA, how else could you start up at full rate? If you had to produce an intermediate, it would take a while to produce enough of it, and more of it, and more of it. And so instead of a straight line, you should get a curved line, but you get a straight line. So Art concluded that there was a molecule that transmitted the information, but it must be so unstable that it must reach a steady state concentration so fast that you can't see that even happening. The paper is totally neglected. It's not cited by anyone at the Pasteur, ever. And the odd thing, he also did the famous experiment with the P32, showing that when P32 wrecks DNA, that all protein synthesis stops. The same experiment is what he and Monica Riley did in Berkeley when Art went back to Berkeley. This time it had Jacob and Monod's name on it, whereas the first experiment, which was really the same experiment, was only Pardee. I don't know what was going on. Art doesn't remember what was going on. Art is 94, I guess, now. I see him fairly often at Woods Hole. But there's a whole backstory to this, which is just not known. So now, as to the happenings at Caltech, I don't remember the slightest talk about any kind of competition. Now, maybe Sydney harbored it silently. We talked endlessly. Every night we went to have steak dinner at a place called Morton's, somewhere in Pasadena. Very dark. It was like a bar, but it was really a restaurant. And we were always accompanied by Dick Feynman. Feynman was very interested in these experiments. But I don't remember any talk about any kind of competition. The C13, it's a very heavy isotope of carbon. Where did this come from? I had wanted to get this in order to study the stability of ribosomal RNA in growing bacteria. And because the RNA molecules are smaller than the DNA molecules, the bandwidth in cesium chloride is broader. To get good resolution, you need a bigger isotopic density separation. So that would mean nitrogen and maybe carbon. So I contacted the Atomic Energy Commission. We don't have high purity carbon-13. Chalk River in Canada, they did not. The Japanese had a nuclear industry, they did not. Linus then, my professor was Linus Pauling. I was his last graduate student. After me, I think here's that. So he was just then elected to the Soviet Academy of Sciences, the Academia Nauk. So I asked Pauling, please ask the Russian Academy, the Soviet Academy, for maybe they have carbon-13. So we sent a telex to Moscow, and we got back, there was no email. We got back at telex, yes, you can have a 1 gram of pure carbon-13 in 1 year. We didn't know exactly what that would mean in 1 year. Later I found out what it meant when, at this meeting in 1961, a Russian scientist came up to me and told me what had happened. That is, his lab mate, a guy named Tikhomyrov, was ordered to make 1 gram of carbon-13 for Professor Pauling. And to do that, they had to make a thermal diffusion column through a hole in the roof up and through the next lab, big, tall thermal diffusion. And they made a gram. And this fellow said that he would like me to send him 1 milligram because he wasn't allowed to have any. So that was the son, that was the father of Alex. Alex, my memory has just failed, who's now at Caltech. Warshawski. And the father, Warshawski, and his wife emigrated to Salt Lake City and so on. So that was the carbon-13. Now, after the last day of the experiment, I got on a plane to go to Boston. First to go to the Gordon Conference, and that's where I guess I must have told Jim about the experiment that Sydney had told me about on the telephone, because I left the very day that they went to the beach. I left for 3 reasons. One, it seemed to me it wasn't going to work. 2, I wanted to go to the Gordon Conference. And 3, I wanted to propose marriage to my first wife, which I did successfully. So when I got back to Caltech, Dick Feynman was still very interested in the messenger RNA. And I think it must have been evidence that I didn't know what Jim and Wally and Francois over in Paris and Howard Hyatt were doing, because Dick Feynman and I were doing the same thing, except in a rather hilarious way. We grew large volumes of E. coli. We had huge amounts of P32. This was in tris buffer, so we didn't have dilution of the phosphorus. And we poured the growing bacteria, which had been exposed to the very hot phosphorus for a very short time, over ice, cracked ice to cool us, and azide, the same thing. But our interest wasn't very deep. Our interest was we weren't looking at ribosomes at all. We wanted to get a hold of this RNA, because we already had concluded from the experiment with Brenner and Jacob that there was a messenger and all that. We just wanted to study it. So it was a double-stranded, stuff like that. So Dick Feynman and I did this day after day in the cold room. And we put this broken up bacteria on the top of a sucrose gradient and drip, drip, get it out. We never got very much. And Dick asked a question that never occurred to me. Are you sure that everything you put on the gradient comes out when you drip it? Well, of course, why? Conservation of mass, in other words. He's a theoretical physicist, the best theoretical physicist around. So it turns out it sticks to the walls of the centrifuge tubes. Caltech had a great machine shop, so we thought, okay, nothing sticks to Teflon. We'll have tubes made out of Teflon, and we'll collect the tubes from the top with a plunger and a little a little Teflon tubing. It sticks to Teflon. So then we had stainless steel tubes made by the Caltech shop, which could do anything in those days. We don't have shops anymore. Maybe Caltech does. Harvard doesn't. Anyway, well, the physicists do, but the biologists used to have one. For saving money, the deans got rid of our shop. Anyway, it sticks to stainless steel. But finally, we got the idea, we'll add something that will stick to the walls, and now the ribosomes, or rather the RNA, this messenger RNA, won't stick to whatever we're going to stick onto the walls, which was gelatin. That works perfectly fine. Then everything comes out. But by this time, I don't know, maybe I was getting ready to go to Harvard. Maybe Dick got sick and tired of it. I don't know why we stopped, but he was a terrific experimenter. He went on to do other experiments. He discovered suppressors in bacteria, suppressors of bacteriophage mutants with Bob Edgard, and he published it in the Caltech student newspaper. So, let's see. One could go on and on and on. And the real pity is that what the playwright and the actors are trying to do, the people who actually should be doing it don't do it. I don't know why. They should. Maybe over drinks they do, but there's no record. So, yeah. Period.
Thank you, Matt. So we have time for just a couple of questions if anyone wants to ask our panelists. Yeah, Keith and Jim, if you'd like to come back up. Jim, no? Okay. Anybody interested? Yes.
So you want to know about the LSD?
I'm just curious where the house was with the doctor with the
So
LSD.
it was just up the hill up here. I never went there. So this is one of the many stories that Sydney used to tell in the coffee room. And actually when I called Sydney about the play and picked his brains and he talked to me for, 45 minutes, I reminded him of that. And actually, I think he'd forgotten that. And then he said, oh, yes, we were doing that. And, I don't think they realized what they were doing because it was 54 or 55 or something like that. But it is documented that the CIA was actually sponsoring the doctor who was on the hill. Yeah. So that was all true. That was all. He wasn't from the lab. Jim, do you know what the doctor who was doing the LSD experiments up on the hill? What was his name? Was like Abrams or Abramson or something.
Up on the hill.
I don't know.
Yeah.
Just two comments. The play was rather interesting and excellent, and it had many of the necessary elements. It had sex. It had drugs. The only thing it was missing was rock and roll. And there is a parallel, at least the way I see it. The microbiologists, people that are doing all of this excellent work, many, many, many struggling to go forward with their imagination, their experiments. And it's very similar to those that are trying to make a career in music. There are many, many, many people pursuing it, very few that ever get recognized. And that's unfortunate. Thank you.
I should say something about the magnesium.
Yes.
Okay, so before Sydney and Francois came to Caltech, I was doing experiments to ask the question of do the 50 and 30S ribosomal subunits stay together forever, or are they exchanging? Okay, so to do that, I was going to do the obvious experiments with heavy and light. And what I observed was that in sucrose and sucrose gradients, 0.01 magnesium was enough, as Jim and Tissier had shown. So I just took that data to stay together. If you lower the magnesium enough, Jim and Alfred had shown they come apart, but at 0.01, which is a lot, they don't come apart. Now, I reasoned that cesium chloride solution at 9 molar, or whatever it was, 8 molar, was actually a weaker salt than sodium chloride. And there's a good chemical argument for that. And they don't come apart at that salt strength unless you put them in a centrifuge. I think what must be happening is that in the centrifugal field, there's an equilibrium of going back together on and off. But if you're in a powerful centrifugal field, the proteins, which are lighter, go up to the top to the meniscus, and up there they're at a totally different concentration, and they can't reassemble. And even if they did, they can't go back and find the RNA cores, which are sitting up here in the gradient. In other words, it was a centrifugal field. We didn't do any experiments to really prove this rigorously, but I think that was the culprit. So,
interesting.
The reason that Sydney thought about magnesium, I'm told by Sydney at the time, which was long ago, Hildegard Lamfrom had made a picnic lunch, and we went to the beach. They went to the beach. I was on the airplane by then to have the picnic lunch, and she told them there about her work, which was to synthesize globin in vitro, and she was explaining that it takes a lot of magnesium to make it work. And it was at that point that Sydney jumped up, but I wasn't there. This is only what Sydney told me as I remember it. So.
You alluded to maybe the messenger RNA didn't get a Nobel Prize because there were too many people.
Yes.
But what about this idea that Sydney alluded to, that maybe this paper that Jacob and Monod wrote after, which incorporated this idea of a messenger RNA, maybe that kind of took some of the, which was published around the same time, right?
Yeah, they don't mention our experiments or Jim's. They knew all about it. But by the word, you mentioned, where does the word messenger come from? You'll be amazed where it came from. In the famous pajama paper, which is about the action of the repressor, the word messenger appears. Not to the transmitter of information, to the repressor itself.
Oh, yeah.
God knows why.
Yeah.
Maybe they didn't know the English well. I don't know why. But that's where it first appears,
Yeah.
is the repressor. Go back and look. You'll see it there.
Okay, other questions? Okay, well, we'd like to thank everybody, and especially our guests, our panelists, wonderful play, and Mila for putting this together.